So we're going to start, if I can see if our speaker is here. So welcome to the last meeting of the calendar year. It's, from that point of view, a special occasion. It's also a special occasion because you have readjusted your basal ganglia and come a week late. Congratulations for managing that. It normally causes great confusion. And it's an especially special occasion because our speaker today is one of our own. Maggie Zellner has been a member of this group, I think, since 1993, 2000. Oh, okay. She's been a member of this group for as long as I can remember, and is in fact the president or the chairman or the director or something of the Neuropsychoanalysis Foundation, which is the financial bedrock of what we're doing here, and also the developmental and educational bedrock of what we're doing here. But she's special not only because she's one of our own, she's also special for, like, probably this is the reason why you qualified to become one of our own, because you're so special. She, very unusually, trained first, well, her first degree was in the humanities, in English literature, semiotics. Okay, it's sort of the scientific end of the humanities. Then she trained in analysis, stumbled upon our field, and then, which shames all of you who didn't do this, she then realized that she needs a training in neuroscience. So having been through the semiotics and the psychoanalysis, she then went back to school and took a PhD in neuropsychology, and she's now doing a postdoctoral fellowship at Rockefeller in this great city of yours, and she's doing research, basic neuroscience research, more or less on this topic, on drive in little humans called rats. And it gives me great pleasure, therefore, to invite up to the stage Maggie Zellner.
I work with mice now. They're smaller versions of rats. So, it's so good to be here, and I'm just going to jump right in. So first I want to tell you what my talk is. It's sort of an educational talk. It's going to be kind of looking at some clinically oriented hypotheses, and I think of it as an example of materialist metapsychology. Because that, for me, is sort of what neuropsychoanalysis is at its heart. Sort of looking at what is the machinery that's going on as we're having our experience, as well as all our unconscious processes that we don't have conscious access to. So it's sort of a different kind of talk than what we normally have here. I'm not going to be presenting a lot of my own data, though I'm going to talk a little bit about some studies that I did for my dissertation. And what I'm definitely not doing in this talk is going into a big exegesis about different conceptions of drive and Freud in 1915 versus 1923, and is there a drive for aggression. I'm focusing on libidinal drives in this talk, and I'm not going to talk about whether aggression is a drive, mainly because for some reason that question mobilizes my aggression. So I don't want to get all irritable with you, so we can leave that for the discussion. And by the way, we're leaving the house lights up because I plan to sort of gesticulate a lot as I'm describing things, so I want you to be able to see them. So as you all know, and also I'm assuming most of you guys are familiar with basic psychoanalytic concepts of drive and related concepts, so I'm not going to go into a lot of definition, though there are some non-analysts in the audience. So Freud talked about drive, this sort of classic definition, as a measure of the demand made upon the mind for work in consequence with its connections with the body. So basically what's going on in your body, and you have needs in order to survive. They get registered in your mind in some way, and then you go out into the world and you do stuff because of that. And it's interesting from a neuroscience point of view, the definition is actually very similar. So this is a sample definition from Roy Wise, who's a leading neuroscientist on addiction and reward learning. And he calls it the energizing effects on behavior of internal stimuli associated with tissue need or hormonal level, or of external stimuli associated with past rewards, in other words, incentive motivational stimuli. So you can see right there that there's a very close resonance between the psychoanalytic conception of drive and the neuroscience conception of it. Then in the Freudian model, there are these different components of drive, right? So there's the source of the drive, what's going on in your body that gets registered somehow. The aim of the drive is to achieve satisfaction and to reduce tension. The object is the thing that you think is going to meet your drive needs, and pressure is the amount of force that that particular drive is exerting at any given moment. One way of looking at motivation, there are different definitions of it in behavioral neuroscience, but I like this one from Yoram Yerbel's review paper from last year called Is There a Drive to Love, which is a really nice review paper. And he says that motivation is a drive that has an object. And I just think of it colloquially, and I think that we all have a sense of motivation is basically feeling like doing something. You are motivated to do something. So it's essentially kind of a motor experience, and it's also a mental experience. So you're oriented towards something in particular that you're wanting. And then cathexis is the investment of libidinal energy. And I know that some of you might not be familiar with that term. So I'll say a little bit more about that in a moment. But I think that the definition of incentive salience, which is the term commonly used in behavioral neuroscience, is actually very similar. And just by the way, I didn't give my whole talk in the handouts, but if anybody missed the handouts, they're on the back table. But I'm very happy to share my entire PowerPoint, so don't feel like you have to madly take notes on it. So the concept of incentive salience, as you can see in this from a piece by Kent Berridge and Terry Robinson, who are sort of leading neuroscience workers on this question. Incentive salience transforms the brain's neural representations of conditioned stimuli, converting an event or stimulus from a neutral cold representation, oh sorry, I didn't edit that correctly, into an attractive and wanted incentive that can grab attention. But incentive salience is not merely perceptual salience. Its attribution transforms the neural representation of the stimulus into an object of attraction that animals will work to acquire. So you can see again that our psychoanalytic conception of cathexis is very close to incentive salience. And I think that the interesting thing about looking at these definitions together is that it's both sort of a property of the object, but it's also arising from the subject. You know, I cathect an object, it has incentive salience for me, but essentially it's my mind and my brain that create that incentive salience. And I think that happily behavioral neuroscience is starting to move away from radical positivist behaviorism, because in the old days it would have been described in terms of it being purely a property of the object. And I think that we have a much clearer sense now, and I hope to illustrate that for you later on, that there's a whole incredible system that is really a dynamic system so that we are attributing meaning to objects. It's not just that we are responding to objects and learning about them. And just to, wait, before I say that, just as long as because I know that there are some non-analytic people in the audience, I just wanted to illustrate. So I think that everyone can get the idea of drive and motivation, but cathexis, so I brought an illustrating object, which I have cathected. So this is, you know, from this great coffee store called Joe on 13th Street. And I also love Starbucks, it's my home away from home, but Joe is really cool. And it's shiny and red, and I carry it around with me all the time. So I have invested my libidinal energy in this particular object. And when it's around, I feel secure, and if I feel for my pockets and I realize I've left it at home, I feel a little bit of separation distress. And then it's sort of multiply determined, right? Because I can look at the cup and I can think of how cool I am because I don't use paper cups like all of you guys. And there's some other meaning that I can't remember at the moment. So I have cathected this object, right? And also I think one of the cool things about cathexis, and this has implications for object relations, which I hope to get into a little bit more later on, is that this object now starts to feel like a part of me in a way. I identify with it, there's something about it that has me-ness in it, you know? And therefore it is more important to me than other cups of its kind. So I think that illustrates the concept more or less. And I just wanted to acknowledge all of these people who have influenced my thinking on these topics. One of those names you might not recognize is my former analyst, who is very influential in my thinking about drive and object relations. So thank you to all of the people who are in this audience and who are not in this audience for what I've taken. I've sort of synthesized a lot, but I think at the end of my talk I have some ideas that might be my own. I'm not sure. So we'll see. And also I think that when we're talking about pathology, so often we have sort of an unspoken model of health in the back of our minds, and so I thought, let me just put my model of health out in front, so that's sort of the context of what I'm talking about. And I think that basically mental health involves being able to feel pleasure and also being able to take actions to get our needs met and our wishes met. In order to be able to take actions which are risky, we also have to be willing to tolerate loss. And we have to tolerate making mistakes, which involves loss of the esteem of people who we care about. So that also involves being able to grieve. And I think that when we talk about sort of clinical and psychodynamic aspects of regulating drive and motivation, this is a huge thing. And so many of us don't do things which we would like to do because we're afraid that it's not going to work out and we won't be able to tolerate that. And I think on a deeper level, often that's because of our relationship to our internal objects that makes that difficult. And I think that's one of the things that we're doing in analysis, is sort of helping our patients and ourselves as analysands, that we're acquiring the ability to tolerate loss. And then obviously when you're out there in the world, you also have to be able to inhibit your impulses depending on what the context is. And so sometimes we go in the other direction and we have a hard time inhibiting things that we ought to inhibit, you know, depending on the context. And as I just said, all of this stuff really involves having centrally a good enough sense of self. You know, I will be okay if X person doesn't love me, if this talk doesn't work out, you know, all of those things. And then as I'm relating to you guys in the audience, I'm also relating to my internal objects, you know, representations of people who have been important to me, and am I pleasing them, am I going to get punished, am I going to get abandoned, and so on. So that's, I think, a big piece of what regulates drive and motivation. And I think that motivation is important because when we don't feel motivated, that's a core aspect of feeling sort of mentally unwell, you know. When you do feel motivated and you feel like you can go out in the world and do stuff, you know, generally you feel better. And as I was just saying about the important role of internal objects, and you know, I was going to go into this whole thing about, you know, this just gives you a flavor of the role of cathexis in the internal world. And I just wanted to, this is from Fairbairn's, one of Fairbairn's essays, and the piece I just want to focus on just for the purpose of time is that, you know, this idea of the internal object being split into an exciting object and a rejecting object. And then those objects are cathected, so we are in a relationship with them, and they sort of are drawing part of our ego energy away from being able to interact in a productive way. So I know that I'm talking about sort of high-level stuff in a very simplified way, but I just wanted to give you a flavor of, you know, sort of the role of cathexis in our internal life, and we can talk about this more later. Okay, I think it's always a good idea to start a talk with something that puts everyone in a good mood. So most of you have seen this, but I've seen it 15 times and I'm not bored of it, so I think you'll all enjoy it.
As we have listened to everyone's plans, we have heard what appears to be sounds of laughter. And we studied these for a couple of years without quite understanding that this might be laughter. And then one day we decided to tickle some animals. And we realized that we had to look at the sounds at a very different register than we can hear. So we obtained these transducers that are called fast detectors. They can bring very high frequencies down to our auditory range. And what we did is we could tickle animals and generate a lot of vocal activity that appears to be laughter. These animals would begin to enjoy our company, and they would start to play with our hands, and wherever we would put our hand, they would follow. And when we tested these animals and asked whether they were enjoying this kind of activity, the unambiguous answer was yes.
So I like to make people watch that video when I'm just starting a talk so that your opioid levels have gone up a little bit, and now you're going to think that everything I'm going to say is totally wonderful. Except I just messed up my microphone. Okay. All right, so I think that you can, imagine why I'm showing this in this context. So we can say that those animals, as social animals, are hardwired to seek companionship, to seek contact. There's something about the tactile experience that's pleasurable, from an evolutionary point of view. And then from a cathexis point of view, it looks to me like those animals have cathected Yak's hand, right, when they're chasing him around in the, they know that hand is the source of some kind of pleasure, and they're seeking it out. So that's an example of the kind of a behavioral, and I think you can sort of feel your way into the mind of the rat and what the rat is experiencing. And for anybody who doubts that animals have the same basic feelings that we have, I just want to show you this picture. I think that you can relate to that female Japanese monkey. So they're snow monkeys, and it's in a hot tub. And also, maybe you can also, so she, I mean, it's a natural hot tub. You can see that she looks pretty blissed out there. And they're having a good time grooming. And I think actually we all should be grooming each other a lot more. I think we would feel much better as a society. So that doesn't really have anything to do with my talk. I just want to show you the photograph. Okay, so now we're going to start talking about the brain systems that are mediating some of these processes. And this is a very simple diagram, and if you feel intimidated by it, I don't really understand what your problem is. So I'm not going to go into this diagram yet. I just wanted to have you have a sense of the interconnectedness of these different regions of the brain that I'm going to be talking about. So in the handout, one of the big pages is that graph. And so tonight, you can go home and you can read it, and you can trace through the different lineages and have a good time with that. But I wanted to really highlight that even though in the rest of this talk I'm going to be focusing on specific regions, they're all interconnected. Almost all of the regions I'm going to be talking about are heavily interconnected with each other. And this picture from a really nice review paper is just looking at the cortex. And you can see that almost every node in the cortex is connecting with almost every other major node. These are different major regions in the cortex. So that gives you a sense of just how interconnected this all is, right? So as I talk about this system, the real system itself it's very nuanced, it has detail to it, it's different in different contexts, in the morning it might be a different kind of system than in the afternoon, in golf clothes it's a different system than in tuxedo. But in order to talk about these systems, we have to necessarily simplify it. So the way that I'm going to be talking about these systems is more at this level. And because I have limited time, and because our knowledge of these systems actually is limited, believe it or not, it's really going to be like this. Like little chunks of the system, okay? So now we're going to go on a little whirlwind tour of some of the regions that are involved with brain drive, motivation, and cathexis. But to just give you a three-dimensional brain in your own mind, I want you to imagine a few things, okay? So take a banana in your mind, and then put a little Hershey's Kiss on top of that little twist at the top of the banana. The banana is the brain stem, and the Hershey's Kiss is the hypothalamus, okay? Then you take 2 hard-boiled eggs, put them on top of the banana, so that the little Hershey's Kiss is poking out almost like a nose. That's the thalamus, okay? Brain stem, hypothalamus, thalamus. Then you take a slice of orange, and then a slice of a clementine, you know, the smaller ones, and you put those on the outside of the hard-boiled egg. Then you take a cinnamon roll, you unwrap one arm of the cinnamon roll, you put it on the outside of the orange, and then the cinnamon roll and the orange is the basal ganglia, okay? So that cinnamon roll is the caudate and the putamen, and those little slices of orange and clementine are the globus pallidus, okay? So brainstem, hypothalamus, thalamus, basal ganglia. And then you take a big, big grapefruit and wrap it around the eggs and the cinnamon roll, and that's the cortex. And when you split the cortex in half, there's cortex in the middle as well as on the outside. Okay? So that's the brain. You have the brain in your mind. There's a little recipe, so you need to recreate that for yourself. You can have that. Okay. Here's a nice list of these structures that I'm going to talk about. And I'm not going to read the main functions that each of these regions are associated with, but I'm sort of grouping them as follows. So I'm going to talk about the periaqueductal gray and the hypothalamus as being important for drive in terms of the source of the drive, as Freud talked about. The reticular activating system, I'm not going to speak about other than listing it here, but that's those nuclei in the brainstem that are sending neurotransmitters like acetylcholine, norepinephrine, dopamine, serotonin up into the brain, and that's what Don Pfaff, who's my PI, sort of considers a precursor to libido. It's like you have to turn the lights on in the brain in order for stuff to happen. But that in and of itself isn't really drive. That's sort of a precursor to drive. That's one way of thinking about it. Then I'm going to tell you about the VTA, which projects dopamine connections to the basal ganglia, as well as projecting dopamine to these structures. And I'm going to be sort of grouping this level in motivation, in terms of selecting actions, you know, what you're going to do in a given situation. And then cathexis, I'm sort of putting as the umbrella for the rest of these structures, in terms of what helps you link up rewards and punishments and other cues, and sort of telling you about what's out there in the world and what your experience has been with them in the past and what you're going to do about it in the future. Now I realize this is carving nature at the joints to an extreme level, and if you remember what I was saying about the interconnectedness, that actually cathexis, I think, is involved with activity between these regions, you know, not in a given region. But in any case, these are some of the regions I'm going to speed you through now. Okay, that's sort of grouping them. By the way, those are Mardi Gras colors, so you should know that. I'm from New Orleans, so let's have a Mardi Gras party in February. How's that? Neuropsychoanalysis Mardi Gras party. Okay, so these are the regions I'm going to focus on for the next section, and I think that these are core regions that are involved with feeling like doing something. These are parts of your body that are actually connecting down into your muscles and coordinating motor patterns. And then they feed back into other regions of the brain so that you can feel the feeling of doing that thing or intending to do that thing. So the periaqueductal gray is this column of tissue towards the top of the brainstem, and that is, if you take an electrode and you stimulate along, let's say, the rage circuit, like Jaak Panksepp has laid out. The lowest level that you can get to where you can trigger a full-blown coordinated rage posture in an animal is the periaqueductal gray. If you go below that, there are other motor nuclei in the brainstem where you can trigger parts of the symphony of actions, but you can't do the whole thing. But in the periaqueductal gray, there are different columns in the tissue, and if you stimulate them, you can get, as I said, coordinated instinctual motor patterns, like running away from something, or arching and unsheathing your claws and clenching your teeth, so that when you are getting signals from higher up in your brain, or like incoming signals, for example, like pain, or the feeling of being restrained in some way, your periaqueductal gray is getting at those signals and then initiating these motor patterns. So when you, and I said I wasn't really going to talk about aggression as a drive, but let's say you have the motivation, someone has hurt you and you feel motivated to hurt them. The idea is that you feel pain and your periaqueductal gray is generating an impulse to fight. Now that has interesting implications that I don't know that we've really explored yet in terms of what's going on if your periaqueductal gray is getting some input saying threat or danger, and then it's getting some other input saying you're not allowed to express that. You know, what's happening at the level of the periaqueductal gray? Is it inhibited? Is the firing pattern dysregulated in some way? I don't know that we have any clue about that, but it's an interesting thing to think about as sort of one of the possible bases of intrapsychic conflict. And the feeling that you have, you know, because I think a lot of us in therapy have had the experience of being sort of depressed and feeling inhibited, and then you realize that you're angry about something, but you've been afraid to be angry, and that often is a very effective switch to lift the depression, at least for, you know, those of us who are not severely depressed, right? So it's interesting to think about what's happening down there around that. The hypothalamus, I think most of you have some basic familiarity about that. So here's the little Hershey's Kiss at the top of the banana. And the hypothalamus has different nuclei that are sampling from the blood. So as it's coming through the body, you're monitoring different levels of hormones, different levels of, for example, there are molecules that tell you whether your fat levels are too high or too low, and they trigger hunger, for example. And so your hypothalamus is detecting need states in your body. You know, if you're thirsty, if your salt levels are too high, and so on. But I think also the thing that we don't have a great enough sense of, which is an important piece of this, is that the hypothalamus also has these nuclei that are sending motor patterns down into the brainstem. So this is from a very nice review by Larry Swanson, who's one of the leading neuroanatomists. And he has this great review paper on behavioral control columns and cerebral control of them. So the idea is that this is in a very simplified version of a simple vertebrate brain. You have this nuclei, the paraventricular nucleus, and that's responding to glucose and insulin and ghrelin and leptin, all of these different molecules that have to do with food and need. And then they also can trigger eating behavior. So if you just stimulate that nucleus and the mouse or the rat is in front of some food, it will start to eat. So you have behavioral impulses that are coming out of these nuclei. So they're sensitized. So in the case of sex, for example, if you have a certain level of estrogen in your bloodstream and your estrogen receptors are getting stimulated, or testosterone, that means that those nuclei that are in charge with sex behavior are going to be more stimulated by sexual stimuli. So you have the input to the hypothalamus, and then you have this behavioral output. Now this is interesting where Swanson is noting that aside from the periventricular nucleus, which has to do with eating, the rest of these core nuclei have to do with either reproductive behaviors or defensive behaviors. And that is resonant with how some people have grouped, you know, like when people talk about are there different subsets of drives and how do we group them. It kind of resonates, with Freud's idea of sort of self-preservative drives versus, et cetera, all of those different categories. But you can see that, those are major things to propagate the species and to protect your resources, protect yourself, protect your children so that you can go on and reproduce. Again, libido and aggression, drives. Yes, no, we'll talk about that later. And this is just a more detailed look at the hypothalamus is sending signals to these motor pattern initiators, to pattern generators, and then down into behavior. Sensory inputs come in so that, food, the smell of food. If you're hungry, you're going to orient towards the food, right? If you have a high level of testosterone stimulation and a sexy woman walks across your path and you're heterosexual, you'll probably have sexual thought. Now, in terms of generating sexual thoughts when there's no stimuli around, which would seem like that's a really good indicator of an endogenous drive. I don't know very much about that. If anyone knows about that, we can talk about that in the discussion. And then, as I said, the cortex can regulate what the hypothalamus does, given a certain kind of stimulus. And Marcel Mesulam has said this really nice phrase, which is that the prefrontal cortex frees us from the tyranny of the hypothalamus. And what that means on a certain level is that, as Larry Swanson was saying, if you cut the brain right above the hypothalamus, you have an animal that will eat if you put food right at its mouth. It will mate if it gets mounted, for example. It can even give birth to offspring. But that animal will not do any spontaneous behavior. So the implications are that your hypothalamus is really doing that extremely basic primal monitoring of needs and generating appropriate sort of consumatory or defensive behavior. But you need the rest of your brain to sort of go out there in the world, to seek things, to learn about stuff, to voluntarily emit behavior and so on. You know what, let me just skip this for time. We'll come back to that. So, and this just illustrates a little bit more from the sexual point of view. As I was saying, that testosterone increases the male's activity, so they're just likely to run around the environment more. And then when they encounter each other, if you have appropriate levels of estrogen and testosterone, you'll do these kind of sexual behaviors. Okay, so that was sort of about instinctual motor patterns, and now I want to say a little bit about the basal ganglia, which I think is especially interesting from the point of view of the subjective experience of craving and compulsion, because the neurobiology of addiction is pointing to a part of the basal ganglia as one of the key structures for developing a compulsion to use drugs or possibly to engage in other behaviors. Like if people have addictive video game habits, for example. So here's the basal ganglia. That's a picture of the brainstem and the thalamus and the basal ganglia. If you just look through it as if the brain were transparent, you can look through it and you can just see what it looks like there. And then this is the different sections I was telling you about, right? Here's the cinnamon roll, the caudate and the putamen. And then in there, that's the globus pallidus. You see how it looks kind of like orange slices? So now you have a sense of the basal ganglia in your mind. Now here's the interesting thing about the core aspect of how the basal ganglia works. And this was very helpful to me when I learned about this. So the cortex, the motor cortex, can send signals down into the body so that you do a particular thing. Like I can choose to reach out and pick up my cup. But at the same time that you're having those motor impulses being generated, there are also these simultaneous loops going to the basal ganglia, to the thalamus, and then back to the cortex. So those loops are running, and then there are these also loops going to the cerebellum and back to the cortex. And then this is sort of a cross-section if we slice the brain in the middle, and you can see that it's very complicated, first of all, and there are a lot of inhibitory influences. It's sort of like a set of brakes on another set of brakes on another set of brakes. So the way to think about the basal ganglia in a very simple sense is think of it like the way that an organ works, right? So an organ has all of these pipes that, and there could be air rushing through the pipes. And you have whatever they're called, like the little levers or whatever, that are keeping the pipes closed. If you press the keys and some of those levers are released, the air goes through the pipe and then you hear a sound. So the basal ganglia is sort of like a very complex set of brakes that's regulating motor impulses and then sort of deciding which ones are going to get permitted. It's kind of like a permissions committee or something. You know, yes, we'll do this set of actions right now, and then later we'll do this other set of actions. And the basal ganglia is getting input from the cortex from all over. And it's also getting important information about your need state. So that when you're hungry, you're going to be more likely to release food-oriented behaviors, you know, and so on. So the example I always give when I'm teaching this is that your basal ganglia, especially the dorsal part of it, up here, the dorsal striatum, is involved with habitual behavior. And you don't need a lot of conscious attention once something becomes habitual. Like when you're riding a bike, you're not paying a lot of attention to, now I'm turning the pedals. You just go ahead and you bike, right? So I don't know if you've ever had this experience. So I go into the kitchen because I left something on the kitchen table that I need. And then all of a sudden I'm standing there looking into the refrigerator. I'm like, why am I looking into the refrigerator? I'm not hungry. But as I was thinking about something else, the sensory information came in of refrigerator, and it just triggered that habitual behavior, and then all of a sudden I'm looking into the refrigerator. Or, you know, like for example, I tend to, well, anyway, that's a bad example, never mind. So, anyway, okay, I'll give the example. So I wash my face with soap at night, and I just rinse my face with water in the morning. But in the one morning, I was actually really thinking about a question that I was going to ask Don Pfaff, my PI, and all of a sudden I realized I was lathering up my hands, and it was the same thing. It's like, I don't wash my face right now. That's because I was thinking about something else, and that conscious control was released. The cues were all there, so my basal ganglia was like, well, it's time to wash my face. Okay, so you have a feeling of that sort of the, you know, how much of that is getting run sort of out of awareness, as it were. Now, I'll also say another thing. So, if we think about sort of habitual behavior as one of the key aspects of how it feels to be in the world, you know, when I'm in this situation, I tend to do X. When I see, you know, Starbucks, I tend to go into it, you know, that when I have a craving, my ventral striatum, if I'm in an fMRI scanner, is lighting up. There's something about the basal ganglia that has to do with ways of being and feeling what you want to do, what you're going to do and what you're not going to do. And how much of that is happening sort of out of awareness and unconsciously. So some people, for example, Andrew Gerber and Brad Peterson, in a review article that they wrote a couple of years ago, as well as other people, have said, you know, procedural memory, which the basal ganglia is involved with, is probably a component of transference. You know, in a certain situation, I feel like this, I tend to do this, and so on. Now, I haven't been able to find any neurophysiological evidence for this yet, but if any of you know about it, I'd like to know, look into it. So I've heard that people say if you're golfing with someone and you want to screw up their game, ask them to slow down their stroke and pay attention to it. And they sort of, you know, if you slow down something that's become habitual and you start paying conscious attention to it, apparently it starts to interfere with those habitual programs. So when I heard of that, I thought, well, that's so interesting, because isn't that on a certain level what we're doing when you're lying on the couch in analysis? You're having a feeling, you're having a very habitual way of responding to something. Your analyst says, what do you notice as you're feeling that? And you start to pay attention to it. And there's something about the non-judgmental observation in the analytic process, possibly also in mindfulness meditation and, you know, sort of daily life of paying attention to oneself, that has the potential for dismantling and rewiring some of those habitual ways of being. So again, this is sort of a suggestion of something to look into further, because I've looked for it and I haven't been able to find it so far. Okay. So here's this complicated graph I passed over. But you can see that this is from a review article, and I gave you this graph on maintaining energy levels. So you have the hypothalamus, which is getting input, telling you what you need. Then you have the nucleus accumbens, which is the most ventral portion of the basal ganglia, sort of doing reward calculations and telling you what you should do now and what you shouldn't do. And the nucleus accumbens and the basal ganglia is getting information from all of these other structures. So again, there's that interconnectedness as you're learning about and guiding yourself in the world. And here's just one last piece about the basal ganglia, which I think is interesting in terms of there's a division between the ventral striatum, which is the nucleus accumbens, that's getting a lot of information from the amygdala and the hippocampus and these sort of ventral portions of the cortex. And then as you go back into the basal ganglia, it starts to get more information from the dorsolateral prefrontal cortex and then the motor and sensory cortex, right? So that whole example of like opening up the refrigerator habitually, that's probably because of sensory information coming into this loop. But when you're really feeling motivated about something, especially if it's new or it's extremely exciting, you're going to have a lot of activity here in the ventral striatum. So that's another thing for us to think about in the analytic context, right? That there's sort of a competition as you're starting to form a new relationship with this new object, and how whatever rewards, potential rewards are coming from relating to this new person, and not just an analyst, but any new person in your life. How might that be relating to or competing with your previous habitual learned behavior about other people? Anyway, so I just said that. Okay, so now I'm going to talk about some of these structures in terms of more moving towards cathexis. So at this point, I think most of you are pretty familiar with the ventral tegmental area. I'll say that that's the first area of the brain that I personally cathected. I went to see a talk by Alan Shore, and the VTA seemed to have all this stuff about motivation and learning, and so I really felt like the VTA was a good thing. I started to identify with it. And so it projects dopamine up to the nucleus accumbens and into the prefrontal cortex, as well as to the hippocampus and the amygdala. And natural rewards elevate dopamine levels in the nucleus accumbens, mainly due to VTA firing. So the VTA is sending these cells to the nucleus accumbens. If you encounter natural rewards, your dopamine levels in the nucleus accumbens go up. All drugs of abuse increase dopamine levels in the ventral striatum, and then once you start learning about stuff that's related to those primary rewards, those stimuli also increase dopamine in the nucleus accumbens. So I think that that is one of the key aspects of cathexis. But since I'm just on the topic of dopamine for a second, I just want to say that dopamine does a lot of different things depending on what part of the brain that you're in. And so I think this is especially important for understanding the role of psychostimulants in ADHD, because it always seems counterintuitive. Like if you have a hyperactivity disorder, why would you take a psychostimulant? But that's because dopamine in the prefrontal cortex is doing something different than it does in the ventral striatum. And in the prefrontal cortex, it's sort of helping to regulate the filter of how your neurons are responding to stimulation. So it helps you to focus, it helps you to stay on task. It does a different thing at that level. And then throughout the brain, but especially in the ventral striatum and the amygdala, the dopamine is also important for stamping in associations. So that when your dopamine levels go up, you feel more motivated, you feel more curious, you know, just think about the prototypical person on cocaine, right? And they're all energized and have ideas for projects and they're going to do this and they're going to do that. And at the same time, you're learning about what's in the environment. And you're learning about what did I just do that gave me this nice little boost of dopamine. So those are two of the key things that dopamine is involved with. And you can see it as sort of a nexus of motivation and cathexis. You know, what I feel like doing and how I'm attaching that to things in my environment. So if you take a monkey and you put an electrode into his midbrain dopamine cells and you're recording activity, this is activity from a single cell, and this is lots of different trials, and then as the trials accumulate, you can kind of pile them up on top of each other, and you can see where the activity was the strongest. So this is a monkey who's getting a little squirt of juice in his mouth that he's not expecting, and his VTA neurons are firing. If you play a sound before you give the juice, your VTA doesn't do anything different when it hears the sound. But then you give that monkey a lot of training, over days or weeks. If you give him an unexpected squirt, it will still fire. However, now that he knows that the sound is predicting the juice delivery, now the VTA neuron is firing to the sound. So to me, this seems like one of the absolute core aspects of cathexis, is that the cue now is mobilizing those VTA cells. And also I wanted to say on the topic of dopamine, especially in the nucleus accumbens, is that I think out there in the popular literature it's a little bit misrepresented, that people are talking about the ventral striatum as the pleasure center and dopamine as a pleasure chemical. But I think dopamine, as Jaak has argued, is very much more involved with the seeking aspect of it. The being energized towards something, and that the pleasure, the yum of something, that's more related to opioid transmission. And Ann Kelly has made that distinction, and other people have as well. And I just wanted to show you just a tiny little bit of evidence for that, right? So here's one study where rats are pressing a lever, and then they're getting a little drop of condensed milk. And condensed milk is very delicious, so they're licking it, and imagining having a good time. So as they're pressing the lever, their dopamine levels are going up. Then as they're consuming the condensed milk, their dopamine levels are going down. And this is over a good number of trials, so it's very replicable. Also, if you look at dopamine levels as animals are either just eating food, it's just there, or they have to work for it, then the dopamine levels go up only when the animal is working for it, and especially if it's working fairly hard, and it's not just a super easy task. So I think that there's strong evidence that, that dopamine is involved more with seeking than with pleasure. Though of course feeling motivated is a positive state. So that's one of the things that we have to make a distinction about, I think. And this is just one other example that if you train a rat that when the light comes on he's going to get a sucrose pellet, then if you inject amphetamine into his nucleus accumbens and he'll press the lever and just get the light and not get the sucrose anymore, he will press the lever that is generating the light a lot more than the lever that's not generating the light. So if you think to that, crazily brief thing that I just went over about Fairbairn's exciting objects, that's kind of an interesting thing to think about. If you have an internal representation of this object that's so alluring and is finally going to give you everything that you want, if only you're perfect, or if only you achieve X, or whatever, then it's interesting to think about how unconsciously are you sort of producing dopamine surges in relation to that particular object, and it's sort of making everything else sort of fade out by comparison, unconsciously. So again, that's sort of dangerous to make those kinds of level transpositions, but we can talk about that later. And then also, just so that you have a sense of what the nucleus accumbens is doing, it's not like they're all firing when you get a reward. They fire, some of them fire, for example, this is a neuron that likes cocaine, quote unquote, right? It fires to cocaine, it doesn't fire to receiving water. Here's a different experiment where you have nucleus accumbens, some of the cells fire when the animal is doing something, some of them get inhibited. Some of them don't do anything when they get drugged, but they will respond to a drug cue, but this neuron inhibits to a drug cue. So again, if you think about that complex set of breaks in the basal ganglia, they're going to be doing different things at different times, depending on the context. It's not just all like, oh, let's boost our dopamine levels and we'll be happy, because your basal ganglia neurons are doing complicated things. And then in terms of sort of sexuality and attachment, there's a whole discussion about are there different drives for sexual behavior and for attachment behavior? How much do they overlap? So I'm not going to go into that today, but this is just one of those key pieces of evidence about the role of dopamine in partner formation. So these are these really cute animals called voles. And if the vole has a lot of vasopressin receptors, which in the male sort of plays the same role as oxytocin in the female, though females and males have both oxytocin and vasopressin receptors. But if it has a high level of vasopressin receptors and it mates with a female, I'm not sure which one is which yet, then they will form this monogamous bond, this particular strain of voles. If you're a meadow vole and you don't have that many vasopressin receptors, you mate, but you don't form the partner bond. And the reason why both oxytocin and vasopressin and dopamine are important for that formation is that if you block one or the other, the animals will mate, but they won't form a connection to each other. I mean, a bond. Now, if you inject it's a virus procedure that allows you to inject additional levels of receptors into a particular part of the brain. And if you make a meadow vole be more like a prairie vole with increased vasopressin receptors, then it will form partner bonds when it mates. So, you know, there's a whole literature on that that's evolving, but I think that it's still not clear yet the extent to which sexuality and attachment are sort of branching from the same drive. Are they really different circuits? How they interact? I think that it's still a story that remains to be sorted out. Okay, so the amygdala is important for forming cue stimulus associations. We know about the amygdala mainly from fear conditioning. I think out there in the world, you know, that if you hear a car horn when you're in a car accident, the car horn will start to be like a scary stimulus. Whoops. Wrong way. But also the amygdala is forming associations between Krispy Kreme donuts and the Krispy Kreme donut logo. So if you've ever had a Krispy Kreme donut, they're totally delicious, and then they make you feel sick about a minute later. But apparently that first burst of deliciousness is enough for your amygdala to form a pretty strong association, so that when you walk by a Krispy Kreme donut store, it's hard not to go in. Though I do manage to resist it. Now, it's interesting because you can learn, there's evidence that you can learn fear associations just by watching someone have a fearful experience. Like if you see someone get a shock when they see a blue square, you will start to have a fear response to the blue square. So that's some work by Elizabeth Phelps. Now I don't know if anyone's done this in terms of reward learning, but I think that if it works for fear, it probably works for positive conditioning as well. You know, so is that part of how we learn what to value, for example, from our families? You know, we see our families valuing certain things, and therefore we start to value them. And we can value those things in very passionate ways. So usually I find that Krispy Kreme thing distracting, so I replace it with this other logo, which is meaningful to certain people. Okay, now this is where I think it gets really interesting. So now we're looking at the orbitofrontal cortex, which is getting dopamine projections. It's also getting a lot of sensory information, so that you're starting to represent the sensory aspect of something, like a taste, and also the reward aspect of it is really getting integrated together. So here's an experiment where the monkey is seeing these symbols, and he's learned the connection between particular symbols and particular treats. So he appears to like apples more than cereal, because when he sees the signal saying you're going to get, he gets presented with the signals, and then when he gets the treat, when he gets cereal, his neurons respond a little bit, but when he gets the apple, his neurons respond a lot more. However, in another trial, we can now see that he actually even prefers raisins over apples. Because when he gets the apple, you see a small amount of activation, nothing like what it was there. But now when he gets the raisin, that's when he gets the big burst of activity. I think that that's also very interesting from a psychodynamic point of view. And I want to just sort of put that into a personal example, right? So let's say I'm given an opportunity either to watch a football game or to have a really good discussion about object relations. Which one is going to be more rewarding to me? Well, I think that I will choose the object relations discussion over football. However, if I have a choice between an object relations discussion and an afternoon with George Clooney, wow, that's kind of a tough discussion, but I think I would still go for the object. That's a little bit of red herring. I really think I would choose an afternoon with George Clooney. So if George Clooney ever watches this lecture on the web, I'll give you my contact information. So is that part of what we talk about in psychoanalytic theory in terms of mobility of cathexis? You know, there are certain things that are rewarding in one context, but they're not so rewarding or motivating in another context. Now, another way that this might relate to psychodynamic activity is that let's say you have a goal, like working on a talk that you're given. And there's certain rewards, but there's also certain punishments that might arise if people don't approve of your talk. And let's say you have an internal representation of someone who's important to you who might be critical of your talk. Well, in that case, it might become very, very rewarding to watch more episodes of The Office. So even though you know you should be working on your talk, it's really compelling to watch that next episode of The Office, even though you've already watched it 3 times. So I think this could be also part of what's going on in the brain when we are sort of managing impulses towards some rewards, but there's dangers associated with it. So instead of going for X, we start going for Y. And if we're not conscious of what's keeping us from going after X, we might just feel like, well, hey, Y is just more rewarding. That's just the thing that I want more. But if you, through an analytic process or some other kind of, mindful practice, can start to understand what's keeping you from understanding what you really want and what you have to do to get it, then I would predict that your orbitofrontal cortex neurons are going to be operating in a different way. This is a really intriguing thing about the orbitofrontal cortex that I think also is relevant for psychoanalytic thinking. It's a recent paper by these guys at Harvard, I believe. And I found it difficult to summarize this, so I'm just going to read from the abstract and quote a couple of pieces from the paper. So this is about how affect is integrated with perception. So it's saying people see with feeling. We gaze, behold, stare, gape, and glare. In this paper, we developed the hypothesis that the brain's ability to see in the present incorporates a representation of the affective impact of those visual sensations in the past. This representation makes up part of the brain's prediction of what the visual sensations stand for in the present, including how to act on them in the near future. So right away you think transference. I'm in a certain situation, here's a person, how am I inclined to act right now? The affective prediction hypothesis implies that responses signaling an object's salience, relevance, or value do not occur as a separate step after the object is identified. Instead, affective responses support vision from the very moment that visual stimulation begins. So this paper is it's all based on construction of visual perception. And I'm going to quote a little bit more from the paper because it's just so interesting. The orbitofrontal cortex is a heteromodal association area that integrates sensory input from the world and the body, in other words, from extra and interpersonal space, to create a contextually sensitive, multimodal representation of the world and its value to the person at a particular moment in time. So am I going to watch football? Am I going to read a book about object relations, right? And what's going on inside of me? What are my needs? What are my fears? All of that is converging in the orbitofrontal cortex. Then, this is very interesting also. Through strong projections to hypothalamic, midbrain, brainstem, and spinal column control centers, the medial orbitofrontal cortex uses this low spatial frequency visual information to modify the perceiver's bodily state, to recreate the affective context in which the object was experienced in the past, to allow subsequent behavior in the immediate future. Now, these authors are very careful to say that this is their hypothesis. This is a model that they're developing based on a lot of evidence. But I think it's, there's very, very good evidence for it, apparently. And just to go into just a little bit more detail, this prediction is part of the brain's preparation to respond to an object based on this gist level prediction, even before the object was consciously perceived. So if you think for a moment about what Joe Ledoux talks about, right, that there's sort of this quick, it's in a different domain, but it's a similar process, right? In the amygdala, you can get quick information that doesn't go to the visual cortex about the general shape, let's say, about an object. And then you get visual cortex information that's more elaborated that then also goes to the amygdala. So if you see a curled up shape on the road, you jump back before you even realize that you saw anything because your amygdala is getting the signal at a very low resolution, it's very rough, grainy image, quote unquote. Oh, snake. So it just generates the whole fear response. And then the conscious perception comes in, you say, oh, that's just a curled up old piece of rope. And then the cortex can then inhibit the amygdala, and also inhibit the HPA axis, which has gotten triggered, and so you can sort of calm yourself down. It is represented as a person's reaction to that object. So, actually, I've already said, clear implications for transference, that you're responding to this person in the moment based on expectations from the past. This piece right here, I think, is very interesting in terms of what we work with around making distinctions between the self-representation and the object representation, and especially what we've talked about in terms of projective identification in psychoanalytic practice. So in a nutshell, projective identification is, for example, I'm feeling angry about something, but I'm afraid to acknowledge my anger. I'm feeling the anger, but I can't own it. So the anger must really belong to someone else. So I project it into my boss. And then I imagine that my boss is angry at me, and then I'm all busy trying to figure out how I can manage him being angry at me. That's one part of projective identification. And furthermore, I might start acting towards him in a way that actually makes him angry with me. So that's sort of the double loop of projective identification, right? I'm feeling something, it's my feeling, but I project it into the other person. Then somehow I can also make them identify with it and then start to treat me in that way. So it's a very complicated, but I think, in my experience, a very real process that's going on inside of us, and that it's extremely important to work through that in analytic work. And so this is a very interesting thing, right? That in the orbitofrontal cortex, as you're generating the feeling about the object, it might be hard to make the distinction. Is it the object that's scary, or am I the one that's feeling scared, for example, right? Just to put it in very simple terms. So anyway, and the rest of that, as you can see, that just sort of covers the same ground. So in any case, that's an interesting implications for a psychodynamic process, and the overall implication being that cathexis is shaping our perception. So based on what has been rewarded and punished in the past, is already guiding sort of how we're attending to things. Am I expecting to have a good time when I pick up my red Joe cup? That's part of my actual perception of the object. And then I didn't really know where to stick this in, but I just wanted to say that in terms of feeling motivation, the experience of something being meaningful, it is also involved with activity in the insula. And we don't talked about the insula all that much yet, I think, in neuropsychoanalysis, but it's an important region because this is just a summary from a paper by Bud Craig where he's looking at all of these different subjective experiences that are related to anterior insula activation. And especially, one of the interesting things is that when people have an aha moment, when they recognize something, there's activity in the anterior insula. And that feels to me very much like in the analytic process, you're sort of listening with the third ear, as Theodore Reich said. You're kind of attending to stuff, you're looking at stuff, you're trying to move back and forth between paying attention to how you're feeling and what you're thinking about. You're doing that within yourself, the patient or the analyst is also doing that, and then all of a sudden something comes together and you say, wow, I understand this now. And that the understanding together with an emotional feeling is a key piece of what is transformative in psychoanalytic process. So something about this node where subjective experience is coming in and recognition and then feeding back to these other structures, I think is important for working through also. Oh my God, how much time do I have left? Okay, so I kind of have lost track of how much time I have left in my own talk, but I just want to say just a quick thing about the acquisition of cathexis for anybody who's interested. Remember that neuron that starts to fire to the sound and not the juice? How does that neuron know when to start firing? So this is what my dissertation work was about, and my mentor, Robert Rinaldi, had this proposition that the VTA cells start to fire to the stimuli because the stimuli gets associated with a reward signal that's coming in at the same time, and basically through this process called long-term potentiation. So I just wanted to, for any of you who are really interested in how this happens, this is a very basic explanation of long-term potentiation. So let's say you have a VTA neuron here, and you have a glutamate neuron, and let's say the glutamate neuron is carrying some information about the light in this chamber. You're a rat, pressing a lever, the light comes on, and you get a food pellet. So the light is coming in, but it doesn't really mean anything. And on a physiological level, you have a glutamate release, but it's a very weak stimulus, and this cell doesn't fire. Let's say the animal eats a food pellet, so it gets a reward signal, and we summarized evidence that acetylcholine in the VTA is a strong candidate for that primary reward signal. So the reward signal comes in, that's a strong stimulus, so this neuron is depolarized, so it fires, and then you have an influx of calcium, so the internal condition of the neuron now is changed. And then when that light stimulus comes in, because of these changes here, there are longer-term changes where you can get more receptors, or the ion channel strength will change. And so now, because you have these changes in the synapse, when that same signal comes through, now it can activate the VTA cell in itself. So that was our hypothesis. And then I just have to show you, this gives me the street cred as a behavioral scientist. So even if this particular set of experiments isn't the most fascinating in the world, at least it proves that I have some credentials. So here's the rat. He's pressing a lever, he's seeing light, and he's getting some food pellets. And what we did was we injected an NMDA receptor antagonist into his brain called AP5, because the NMDA receptor is considered one of the key mechanisms for long-term potentiation, because when the NMDA receptor opens up, you get calcium, and that facilitates all of those changes that I just showed you. So if you inject that right into his VTA, in the first 4 sessions, if he got no AP5 or a very low dose, he learned to press the lever for food normally. But the rats that got a higher dose of AP5 sort of seemed like they remained agnostic, like they couldn't learn to press the lever to get the food, and we did controls that they were pressing the lever, they were eating the food, but they seemed not to be forming an association. So from a psychodynamic point of view, we could say they were not yet cathecting the light or the lever. Then, when they didn't have any treatment, all of the animals learned normally. And then on their last day, we gave them another injection of AP5, but it didn't bring them back to these levels. So basically, once you have the association, you didn't need the NMDA receptor anymore to trigger the response. Then we have another, we did another study that was just looking at Pavlovian approach. So all the animal, just the light came on and a food pellet dropped in. And then he would have an extinction session where he just was in the chamber and nothing was happening. And then a final test session where he just got the light. And if the light has become a conditioned stimulus, if it's acquired incentive salience, the animal should go check the food trough when the light comes on. And so we injected them with AP5 during the conditioning sessions. And what we found was that if they just had regular saline injected with no drug, they started to learn normally. So they started to actually approach the food trough when the light came on. Then they all had an extinction session. And then when we just gave them just the light cue, only the animals that had gotten saline were approaching the light. So again, we could say that these were the animals that had cathected the light. It became important to them. If they got their NMDA receptors blocked, or they just had random lights and food pellets dropping, they didn't learn. So now your burning desire to understand, the mechanisms of long-term potentiation in the VTA have been stated. Okay, so I'm coming into the home stretch, and then I look forward to hearing, because there's so much that could be talked about on all of these different topics, so we'll see what questions come up in the discussion. Okay, so now I'm just going to say a little bit about the idea of top-down control on drive and motivation. So this is from a review paper by Antoine Becara, looking at especially the impairment in top-down control in addiction. But this just sort of summarizes that you have these prefrontal areas that are and he's calling these the reflective system, areas that are activated when you're reflecting on something, you're having conscious experience, you're thinking about what you should be doing, what you should not be doing. And then the dorsolateral prefrontal cortex, and we all know how important these parts of the brain are for self-control because of our famous friend Phineas Gage, who had that part of his brain blown out. So that just gives you a 3D sense of what I'm talking about at the moment. And so the dorsolateral prefrontal cortex, the ventromedial, and especially the orbitofrontal cortex have inhibitory connections going down into some of these lower structures. So the orbitofrontal cortex has direct connections to the amygdala, and to the hypothalamus, and to the periaqueductal gray. And the dorsolateral prefrontal cortex has plenty of inhibitory connections as well. And also the hippocampus can inhibit the hypothalamus, so that when your HPA axis, your stress response system is turned on, your hippocampus can sort of put the brakes on it. And it can inhibit the amygdala as well. Okay, so again, if we think about that from a psychoanalytic point of view, it's starting to sound like at least some kind of substrate for internal objects and for superego. What you're doing based on what's appropriate in your culture, based on what your particular family found appropriate, and so on. So if you watched TV in the 1970s, you recognize these as the archetypal mom and dad. So, of course, all of the parents in the audience are accepted from this lecture. So none of you are being referred to here. But, so the idea being, that we have experiences about what's acceptable and what's not acceptable. And we internalize that, and then that becomes part of how we regulate our behavior. Or, and what choices we make, and then we can substitute other people. That was totally gratuitous. Okay. So this was some ideas. One of these ideas came up for me in reading a review paper by Lane and Garfield that was in Neuropsychoanalysis a few years ago, where they were looking at, that the activation of these prefrontal areas that are involved with inhibition are also involved with conscious awareness and reflection. And so that's an interesting thing if you think about what's going on in the therapy process where you're trying to allow yourself to become aware of things that you're afraid to be aware of. And there seems to be some link between allowing yourself to experience something and be conscious of it, and at the same time increasing your ability to regulate yourself. And so they were making the argument in that paper that there's something about the simultaneous engagement of feeling something and paying attention to the feeling of it that is facilitating growth. And then I had already said that thing about the, screwing up your stroke by paying attention. So I think that there's something about paying attention that is helpful in starting to rewire some of these habitual ways of responding to cues. Now, just briefly, this is some summary from a review paper by Zecchi, where they're looking at these imaging studies of people looking at pictures of people they're in love with versus friends, or their children versus other children. And there's that whole controversy I was mentioning about, the extent to which they are the same or different drives. And it seems to me that in looking at these kind of studies and also the literature on addiction, that ultimately it seems like there's really very similar circuitry involved. Whenever you're talking about responding to emotionally meaningful stuff that you feel motivated by, that you feel rewarded by, that generally speaking you're talking about the regions that I've outlined in this talk. And that there may be subtle differences that we can't pick up yet at the level of neuroimaging. But ultimately they engage, broadly speaking, very similar networks. And this is looking at it from a different point of view, which is a review paper that was just looking at people responding to different kinds of pleasurable or painful things, like tasting chocolate, listening to music, or feeling pain, or seeing someone else feel pain. And basically all of the structures that I've mentioned today, going from the brainstem, all the way up into the prefrontal cortex, that they're activated by both. So it's not that they're separate circuits for pleasure and then other circuits for pain. There seems to be these circuits for basic well-being and then learning about cues and making decisions, and that those are activated both by pleasure and pain. And I think that we can think about how that relates to our understanding of the psychic life and psychoanalysis.